Welcome to Learning Management System, Commissionerate of Collegiate Education, Vijayawada, Andhra Pradesh. Myself, Indravati, Assistant Professor in Biotechnology, KVR Government College for Women, Autonomous, Kurnool. Dear learners, the subject recombinant DNA technology is made easy for you. Do follow all my video lessons to cover different topics related to this subject. Dear learners, today we will be talking about hybrid vectors, that is Cosmids, Phagemids and YACs. And in this session, you will be knowing the following things. What are hybrid vectors? How Cosmid is used as a vector in gene cloning experiment, about the Phagimid vector, YAC vector, that is yeast artificial chromosome vector, and the advantages and limitations of these hybrid vectors. Dear learners, let's study about the hybrid vectors in detail. So, these are the new generation vectors which are genetically engineered to have qualities of more than one vector. You know that the plasmids, they are able to clone only small sized fragments, and if you increase the length of the DNA insert, the efficiency of cloning decreases in plasmid. And if you take lambda fast vectors, there is a size limit. You can remove the non-essential region and only up to 25 kb of the foreign gene can be inserted. So in order to meet the requirement and also to accommodate large DNA fragments, these hybrid vectors, they are designed. So of the vectors, the first one is the Cosmids. So these Cosmid vectors, they were first described by the scientist Collins and Horn in the year 1978. These Cosmids are small sized vector molecules. The size is ranging from 4 to 6 kb, and it can hold DNA fragments of up to 47 kb length size DNA fragments. And this Cosmid, it is a hybrid vector. It is containing cos region from the lambda phase, and the origin of replication, it is taken from the plasmid. Due to this reason, cos plus mid, cos is taken from lambda phase and mid means the origin of replication, it is derived from this plasmid. Apart from these two regions, it is also having polylinker sites, then marker genes, and these Cosmids, they are extensively used in this genomic library construction. So this is the way in which the typical Cosmid it appears. So origin of replication derived from the plasmid, then cos site, it is derived from lambda phase and the antibiotic resistant genes, and also it will be having a polylinker site. In this polylinker site, you will be inserting our desired gene. Up to 46 kb of the foreign gene can be inserted. So these recombinant Cosmid DNA, that is the Cosmids containing our DNA insert, they are packed in vitro as phases. Then these recombinant lambda phases, they are used to infect the E. coli cell. So that is what is seen here. So this is the this is the Cosmid vector, and by breaking the polylinker site, you are inserting our foreign gene. Later, this recombinant Cosmid vector containing the DNA insert is packed into the phase particle, and these phases, they are used to infect the E. coli cell. So the Cosmid infects like a phase, and once this particular phase, it injects the DNA, it circularizes, because due to the presence of the cohesive end, it becomes cos site and the circular DNA molecule is formed. And because this particular Cosmid is having origin of replication of plasmid, it replicates like a natural plasmid, and up to 15 to 20 copies of this Cosmid copies, they are produced inside the E. coli cell. Because it is not containing any phase packaging protein, no heads or this packaging is not seen, and you can be selecting the transform cell by using antibiotic selective medium, and the antibiotic resistant colonies, or they represent the recombinant cells which are having this cosmid vector. So in this manner, the cosmids, they are used as efficient vector for cloning large sized DNA fragments. Coming to the second type of hybrid vectors called as phagemids. These phagemids are derived from phases, filamentous phases and plasmids. So if you see the M13 phases, they are generally used for the production of single stranded recombinant DNA molecules, but there is a size limit. Only small sized fragments, that is 1.5 to 3 kb, can be inserted in the normal M13 phases, and also in the plasmids also you are able to insert only small sized DNA fragments. So to overcome this limitation, the phagemids, they were constructed, and these vectors can be, that is, it will be having the advantage of both the plasmid vectors and that of the M13 vectors. So the size of the phagmid is 1.5 kb, and it can accommodate the DNA insert of size from 10 to 20 kb of the foreign gene can be inserted. So these phagmid vectors, they are having high efficiency of transformation, and they are genetically stable hybrid vectors, and because they are having a origin of replication of both the plasmid and M13 phase, they can undergo single stranded replication as well as the double stranded replication. Apart from these or replicons, they are also having phase coding proteins, code protein genes, then selection marker genes and polylinker sites. So they can be replicated in the host cells like plasmids, and it maintains a double stranded replication and high copy number, so that a large amount of the foreign DNA, it can be recovered. So they can also be packaged as single stranded DNA because it is having a phase origin of replication, so they can propagate in the single stranded form, and also because and it can be packed inside the phase head because it is having a package protein. And these particular vectors, they are utilized for sequencing purposes, for site directed mutagenesis, and to generate hybridization probes like this mRNA or cDNA probes can be produced by using these phagmid vectors. The most common examples are this P blue strict vector and lambda ZAP vector. So this is the way in which a typical phagmid looks like. So it is having origin of replication of plasmid, and also the replication of this phase, and the selectable marker gene is there. So it helps in screening the transform cells, a code protein, and the polylinker site where you are cleaving and inserting the foreign gene. Apart from this, it is having regulatory genes to express our desired gene. So in this manner, due to the presence of the replicons of both the plasmid and the Ig gene, when this plasmid replicon, it activates, it promotes double stranded replication. When the origin of replication of phase is activated, the phagmid vector, it switches to single stranded replication, and later these DNA molecules, they are packed by the code proteins to release viable phase particles, and in this manner you will be cloning large sized DNA fragments using phagmid vectors. Both single stranded and double stranded DNA fragments can be produced using this phagmid vectors. Coming to the third type of hybrid vector, yeast artificial chromosomes, they are simply called as YACs. These artificial chromosomes are DNA molecules assembled in vitro from defined constituents that function like natural chromosomes. These artificial chromosomes were first described in the year 1983 by the scientists Murray and Jostak. So these YACs, yeast artificial chromosomes, they will be acting like a shuttle vector, and they can be propagated in two different host cell, that is a prokaryotic cell as well as a eukaryotic cell. The prokaryotic cell here is E. coli and the eukaryotic cell here is yeast cell. So the yeast can exist inside the E. coli as a circular plasmid, and in yeast and this YAC it exists in the yeast cells as a linear nuclear chromosomes. These YACs has the ability to accommodate very large size DNA fragments, that is from 0.2 to 2 MB of the foreign DNA can be inserted in this YACs. So if you see the features of these YAC vectors, so E. coli origin of replication is there. So it allows that is the circular version of the vector to replicate in E. coli, that is it will be replicating like a plasmid, and it is having yeast autonomous replication site, the origin of replication of yeast, which helps to propagate the vector in the yeast cells. And also it is having other segments like centromere and telomere sequence. The centromere helps in regulating the segregation during mitosis, and telomere helps to maintain the chromosome stability. And apart from these, it is having selection markers for both the host cell, bacterial selectable marker and yeast selection marker. These particular yeast artificial chromosomes, they are having, that is the autonomous replication site, regulatory and marker genes. They function only in yeast, but they don't show any response in bacteria. Whereas the bacterial gene segments, that is the bacterial origin of replication, bacterial regulatory genes and marker genes will not function in the yeast cell. So in this manner, that is the particular YACs, it will be acting as a shuttle vector, and it can be whenever it is propagated in a bacterial cell, all the yeast genes they get or in inactive condition. When it is propagated in a yeast cell, the bacterial segment they are getting inactivated. In this picture, let's see how the YAC works. So this is the YAC chromosome which is in circular form. The yellow color, it represents the yeast origin of replication, the yeast selectable marker, the telomeres, the pink color one are the telomeres. The green portion, it represents the bacterial origin of replication, the bacteria selectable marker. So when these circular YAC is digested with two restriction enzymes, so when you first cut it with BAM H1 restriction endonuclease, the circular form of YAC is converted to linear chromosome. Later, this linear chromosome, when you digest it with EcoR1 restriction enzyme, it is converted to two arms, left arm and the right arm, and within these arms you will be inserting our foreign gene. So this is called as the recombinant YAC vector, so which is having our desired gene. This recombinant linear YAC, it is transformed, that is it is transfected to yeast cells. So this yeast cells, the particular our linear YAC vector, it will be propagating, and the transformed cells, they are screened using selective media. In this manner, our desired gene, it gets multiplied in the yeast cell by using its origin of replication, and you will be getting multiple copies of our inserted gene. So let's see the advantages and limitation of the yeast artificial chromosomes. These artificial chromosomes, they are used to express the eukaryotic proteins in the yeast cells as they require the post-translational modification. YACs, they are also used for physical maps, for creating physical maps of large genomes like human genome. And if you see the limitations, this cloning process is very complicated, and it is difficult to isolate the YAC DNA from the host cell, because the host cell chromosomes, they are also of similar size. And other limitation is because you are inserting large DNA insert, they are prone to breakage, and it results in rearrangement and recombination with other DNA molecules present in the host cell. So these are some of the limitations, but The overall these YACs are greatly they are used as efficient vectors for cloning large sized DNA fragments. At the end, let's recap some of the main points which we have covered in this session. So, hybrid vectors or new generation vectors that are genetically engineered to have qualities of more than one vectors. So, you have discussed about the Cosmids. This Cosmid is a hybrid vector containing cos site derived from lambda and origin of replication derived from this plasmid, and it is able to accommodate about 44 kb of the foreign DNA. And the second type we have discussed about the Phagemids. These are genetically stable hybrid vectors, and they are having a M13 origin of replication and a plasmid origin of replication, and it can use to replicate both double stranded DNA as well as single stranded DNA. And in this Phagemids, you can accommodate up to 10 to 20 kb of the foreign genes can be inserted. And the last one is the YAC vector, yeast artificial chromosome. It can exist inside the E. coli as a plasmid, and in case in the yeast cells it will be maintained as a linear chromosome. And these YACs, they are having the capacity to accommodate large size DNA fragments from zero point 0.2 to 2 MB of DNA inserts can be integrated in this YAC vectors. And overall, if you see the advantage, these hybrid vectors, they are used to clone large size DNA insert, but the limitations are cloning efficiency is less, and because these large size DNA, they are prone to breakage. Those are some of the limitations. With this, I conclude the session. Hope you enjoyed learning this session. Last but not the least, I profusely thank Commissionerate of Collegiate Education for providing this LMS platform to reach a large number of enthusiastic learners like you. Thank you one and all.